All right, let's jump right in. Today, we're going to talk about a piece of technology that's, well, it's completely changing how we look at health, disease, and really the nuts and bolts of life itself. We are talking about the protein microarray. I mean, that sounds like something straight out of a sci-fi movie, right? But what if I told you the key to doing just that? It lies in a technology that can scan for thousands of different clues in that one drop of blood all at the same time. That's exactly the promise we're going to unpack today.
But first, to really get why this tech is such a huge deal, we have to understand a fundamental challenge in biology. It's something people call the protein problem.
Okay, so we all hear about DNA all the time. It's the blueprint, the master plan for life. But here's the thing you have to remember. A blueprint doesn't build a house. Proteins, they are the workers. They're the enzymes, the messengers, the scaffolding. They're the ones actually doing the work, carrying out all of life's functions.
And that gets us to the core of the problem. Your body has tens of thousands of different proteins, all interacting in this incredibly complicated dance. Trying to study them one at a time is unbelievably slow. And worse, you miss the entire big picture. You'd never figure out how a city works by talking to just one person, would you? You need that bird's eye view.
So how do we get that bird's eye view? Well, this is where a breakthrough technology comes into play. You can think of it like a biological microchip.
So a protein microarray is, at its heart, just a small glass slide. But it's a very special slide. It's been printed with thousands of individual, purified proteins. Each one sits in its own tiny, specific address, creating this super dense grid of biological information. It's the whole city, mapped out block by block, all on a single slide.
And these amazing little chips really come in two main flavors. You've got your analytical arrays, which are basically just taking attendance. They tell you who's there and in what numbers. Then you've got functional arrays. These are way more interesting. They study what the proteins are actually doing, who they're talking to, what they're binding to. It's the difference between asking who's at the party and what is everyone at the party doing.
Now you might be sitting there thinking, how in the world do you build something so tiny and so complex? And you're right to wonder, it's an incredible feat of engineering and robotics.
Let's walk through it real quick. First, scientists turn cells, usually yeast, into tiny little protein factories, churning out thousands of different kinds. Then, robotic systems take over, purifying every single one of those proteins automatically. And finally, a high-precision robot carefully prints a microscopic dot of each protein onto a glass slide. We're talking about a system that can purify over 1,000 different proteins in a single day. It's just wild.
And this image, this shows you the whole workflow in action. This isn't just a diagram, it's the real deal. You start with genes and yeast, go through those robotic purifiers, and you end up with the final chip, all those glowing dots you see here. This process is so powerful that scientists have actually made microarrays with nearly the entire proteome, that is, pretty much every single protein, from an organism like yeast.
Okay, so we have this incredible tool. Now for the really fun part. What can we actually do with it? This is where we start to decode the secrets of disease and the very rules of biology.
Let's start with the big one, a question that matters to pretty much everyone. For diseases like cancer or autoimmune disorders, catching them early is everything. So how can we spot them long before any symptoms show up?
Well, the answer is to look for a disease's unique molecular fingerprint. See, just like a criminal leaves behind evidence, a disease leaves a trail in your blood. And that trail is often made of specific proteins called autoantibodies. Think of them as rogue antibodies that your body accidentally makes to attack itself. And this is exactly how we can find them. You take one of these human protein microarrays, you wash it with a drop of a patient's blood serum, and then you add a little tag that glows. If that patient's blood has those rogue autoantibodies, they'll latch onto their specific targets on the chip and light up. Every single glowing spot you see here, that's a hit. It's part of the disease's fingerprint.
And this is why scientists are so excited. It is an incredibly powerful, systematic way to find these early warning signs, these biomarkers. What this really means is that we're getting closer to a future where a simple blood test could spot something like ovarian cancer years before you'd ever feel a symptom.
But the power of these chips goes so much deeper than just diagnostics. We can use them to ask some of the most fundamental questions about how our cells even work.
Look at this. This shows a super cool experiment to map out the cell's internal wiring. Scientists took a yeast proteome chip and basically tested which proteins could be flipped on or off by these other proteins called kinases, which act like tiny molecular light switches. In one single project, they found over 4,000 of these connections, creating this massive map of the cell's communication network. And the applications are just staggering. I mean, researchers are using these chips for everything, from mapping how proteins work together, to screening how new drugs find their targets, to figuring out how viruses like herpes actually hijack our cells. They're even using them to profile our immune response to infections like SARS with a sensitivity that's 100 times better than the old methods.
So, this all brings us to the future. If this technology can already do all of this, you have to wonder, where is it all going?
Just look at this number for a second. Over 17,700. In just one experiment with a human protein microarray, researchers discovered that many new interactions between proteins and DNA. That single study literally doubled our entire library of known binding sites for human transcription factors. That's the mind-boggling scale of discovery we're talking about here. So the future, it seems, is all about teamwork. It's about combining that bird's-eye view you get from protein microarrays with the super detailed analysis from other technologies, like mass spectrometry. When you put them together, we are heading towards an understanding of biology with a resolution we've just never had before.
It really makes you stop and think, doesn't it? From predicting disease in a drop of blood to mapping the very circuits of life itself, this tiny little glass slide holds just incredible potential. And it leaves us with one big question. What mysteries of the human body are we going to solve next, one tiny spot at a time?